So welcome back to the Inspire seminar series. I'd like to start by saying that as we gather virtually today, we acknowledge that most of us are on Treaty 6 territory and the homeland of the Métis. We pay our respect to the First Nations and Métis ancestors of this place, and we reaffirm our relationship with one another. So just some housekeeping before we get started. So again, this is the Inspire seminar series. This is a series focusing on synchrotron-related innovation, applications, research activities, or professional developments. This series runs on every other Wednesday from 1:30 to 2:30 on Zoom, such as right now. And if you have any suggestions for speakers, please let us know. So contact myself, Ardalyn, or Lenore, and we will see if we can invite them. So for today, we'll have a speaker. He will be speaking for 50 minutes, and then we'll have about a 10-minute for a Q&A session. And for anyone who's still joining, if you could keep your video and mic off during the presentation, this will reduce bandwidth and distractions for everyone else. If you have any tech issues, please message Ardalyn or Lenore. If you have questions for the speaker, you're welcome to add them to the chat at any time, and I can ask them for you at the end when we get to the Q&A session. Or if you want to wait till the end and then raise your hand, you can ask it yourself when we get to that point. So for today, our speaker is Dr. Derek Peek. He's a professor in the Department of Soil Sciences at the University of Saskatchewan. And today he'll be presenting on shining light on phosphorus in the environment. So Derek, floor is yours.
Yeah, thanks for the introduction. Thanks for showing up. I'm going to talk a little bit about a lot of different research that we've done on the topic of looking at phosphorus with synchrotron techniques. And it sounds simple, but there's all sorts of challenges. And so rather than giving you a presentation where everything works, I thought I would kind of explain how our research program ended up, working on a lot of different things to try to make things happen. So if you're not a soil scientist, then I'll give you a little bit of a background on phosphorus. It's essential for plants and animals to grow. It's a macronutrient. And in fact, the development of commercial nitrogen, phosphorus, potassium fertilizers is responsible for the green revolution and our ability to feed all the people that are in the world today. However, the availability of phosphorus is finite. It's a natural resource that's mined. And so efficient and responsible use of that fertilizer is a major component of global food security and also agricultural sustainability. The flip side of phosphorus is that it's not only a nutrient that is required for life, it also is an environmental contaminant in many surface waters of the world. Over application of biosolids and manures and fertilizers into agricultural fields where you exceed the capacity of the plants to take them up and of the soils to hold that phosphorus ends up producing environmental problems in many parts of the world. And that can include eutrophication, algal blooms, fish kills, and even groundwater issues in some places where phosphorus has not been managed properly. And something that not a lot of people probably have thought about in the phosphorus cycle is, related to that. I mean, what you're seeing is that when you put too much phosphorus out into the environment, then organisms grow and proliferate that you don't want. In many subsurface environments where we've accidentally spilled hydrocarbons and organic contaminants, there are microbes there that could break them down and make the land and help reclaim the land and remediate it. But getting phosphorus to the microbes in the right amount at the right time often will limit the success of removing hydrocarbons from those sites. And so really in all three of these systems, agricultural, environmental water quality, and remediation, the phosphorus chemistry is pretty similar, and it is a function of the pH of the system, of the temperature of the soil solution, of the types of minerals that are present, and of the biological activity. So you could start to partition or speciate phosphorus into dissolved, which you would expect to be the most available for plants and organisms, but also the most susceptible to leaching and ending up in the environment in unwanted places. You could look at phosphorus that adsorbs reversibly on the surface of mineral and organic fractions of the soil, and then also, if the solubility or if the phosphorus concentration gets high enough, then you start to form new minerals, calcium phosphates, aluminum phosphates, iron phosphates, that are going to be much less capable at replenishing that solution phosphorus. And then, yeah, based on what you're doing with that soil system, whether it is growing crops, then plant uptake is obviously important. But even in those systems, microbial activity and mobilization of the phosphorus and mineralization of the phosphorus is going to go on. So if we can really understand what the pools of phosphorus look like in systems, then we can optimize them so that we get the most plant growth with the lowest risk of environmental transport, or so that we get, again, the optimal amount of bioremediation without causing a lot of unintended effects. And so, in fact, then looking at the species that form, they have a large effect on the fate of phosphorus in soil. And that we know this, we can look. This is the Soils Home Study course from the University of Nebraska co-op in 1999. So it's not super sophisticated science, but the trick is that, as I've made this little box around calcium phosphates, those are typically the dominant phase in soils in this part of the world, where the pH is usually between 6.5 and 8.5. But calcium phosphate actually encompasses a wide variety of materials that differ in their solubility by about 12 orders of magnitude. And that runs the gamut from fairly poorly crystalline, fairly soluble phases like dicalcium phosphate dihydrate or dicalcium phosphate anhydrous to poorly crystalline but apatite structured amorphous calcium phosphate to things that are not very soluble at all, like hydroxylapatite. And so I have phosphorus K-edge XANES on the right here, and you can see that we can do a pretty good job in this already relatively complex system, looking at what's happening with phosphorus and calcium just using synchrotron X-rays. So there's been a lot of interest, and I'll talk about some publications that we did back when I was in grad school, back in the dark ages, I guess. And there have been hundreds of papers that have looked at trying to understand the form of phosphate in soils and in manures and biochars and biosolids based on phosphorus XANES. But the problem, I guess, sort of the dark secret of this is while your reference materials all have very different spectra, and you can make constructed mixtures and do speciation and everything looks good, when you actually measure the speciation of phosphorus in a soil, you typically find that there's not nearly as much structure. And as a result of that, and the reasons for that are really that organic phosphorus, adsorbed phosphorus, and really poorly crystalline phosphorus materials all have basically the same XANES spectra in the K-edge. And those forms of phosphorus collectively might be 75 or 80% of the total soil P in many systems. And so under those kind of conditions, I mean, you can see in this data set that there's a little bit of structure beyond just a featureless spectra, but not a lot. And so as a result, what you often find is that you could fit any of these calcium phosphate phases that I was talking about before as a component of this data set. You just change the amount as you change the amount of structure that the XANES of the reference material has. And that's a fancy way of saying that there may not be a unique solution for fitting phosphorus speciation in most soil samples or in many soil samples. And that is not very satisfying academically. And so we've done a reasonable amount of work, I think, in coming up with options to improve. And one thing, I'll start off with the good news. The current detection limits at SXRMB beamline at the light source, and I'm sure that's where I do all my work now, but I'm sure there's other facilities that have similar detection limits. They're good enough. I mean, just the background unfertilized soil sample in this part of the world gives you a reasonable phosphorus XANES spectra in an hour or two of data collection. So you don't have any problems with detection limits, which is good. But what you sometimes have issues with are if the concentration of phosphorus is dramatically changing in your reference material versus your dilute or your control samples, you can have self-absorption or saturation effects that'll distort the spectra. And when you're trying to fit a largely featureless sample, then you're really gonna, the quantitative bit of it is really gonna be affected by that. The other larger problem with phosphorus speciation is one that's not unique to phosphorus. It's that your ability to do any kind of linear combination fitting or quantitative analysis is only as good as your reference library. And the situation in the case of phosphate is that every research group does their own preparation, synthesizes their own phases, and there's not a lot of communication back and forth. And so it's not like you can come up with a crystal structure and generate a theoretical XANES spectra for these materials. And that means that there's a big time investment into doing some of this research. And the phases that you find in the environment may not be pure synthetic phases like you could buy from Fisher or Alpha Azar. And I'll talk about that in a bit too. And then finally, no matter what you do, the aqueous, the adsorbed, and the organic phosphorus standards, they all look very similar to one another at the K edge. And so speciation among that group is going to be fairly limited. But basically what you see is that aqueous and adsorbed phosphate have a much higher peak at the white line than the organic phases. But as I mentioned, that's also subject to saturation and self-absorption effects too. And then the final real problem with this is that almost all the phosphate in the system is +5 and it's surrounded by 4 oxygens. And so very subtle shifts in the position of the edge are all that you're going to see in terms of the energy shifts of the system. But calibration is problematic because of the fact that you're never going to be able to get X-rays through the sample to be able to have an internal calibration standard. And so regular checks with your reference materials are really eat into your beam time if you're going to be careful about this stuff. And if you're not, then what you'll find is that float that you have on your x-axis in terms of the E naught is going to be another fitting variable that makes it very hard to be successful at doing speciation in natural systems. All right, so this is not a super positive situation so far, and I guess as I'm like an old guy yelling to get off his lawn out on the front porch. But in fact, what I'm going to show you from the rest of the presentation are there's a lot of options that you have to deal with this situation. All right, and the first option is just to go after low-hanging fruit. You could study samples that have very strong XANES features. That could mean that if you are interested in the fate of organic fertilizers and manures and biosolids and things like that, then rather than try to study them after they've been placed into a soil and diluted and undergone transformations, study them as the initial material and start to look at how conditions like the diet of the animals, the synthesis conditions of the biochars are going to change the phosphorus chemistry and speciation. And then you can link back to people that are doing field trials and try to make some inferences about how the bioavailability and the structure are related. And I've got a few papers here where we've worked on that over the years, and I'll show you a little bit of data. Initially, I guess my first paper when I was a PhD student, way back in 2002, was looking at issues associated with over-application of poultry manure in the East Coast of the US. That's why I had that picture of all those chickens in my environmental aspects slide earlier. And it turned out that the best way to manage the availability of phosphate out in the field was to manipulate what was in the chicken manure before it went out and was applied. And so in normal, like best practices, they give the chickens a large excess of dicalcium phosphate that's poorly crystalline. And the corn and other grains that they feed the chickens is phytate and phytic acid, and they don't have the enzymes to break it down. And so a large amount of the phosphate that the chickens need just pass right through them into their manure, and then into ultimately into the Chesapeake Bay or into the Atlantic Ocean. And so we did some studies where we systematically looked at changing the diet of the chickens. You could give them less of that calcium phosphate, but give them an enzyme so that they could cleave the organic phosphorus and hydrolyze it and make use of it. And by doing that, you can kind of see from the left side here to the right that we shifted the type of calcium phosphate that formed into a much crystalline phase. And then if you looked at the linear combination fitting, then we also lost the adsorbed or aqueous phosphate. And so we've shifted the majority of the phosphorus in the system from something that's fairly soluble and fairly high risk to transport into a phase that's going to be stable when you put it out in the field. And so making those sort of observations with phosphorus XANES already gave us much better management outcomes. The other thing you can do is try to be more careful and more clever, I guess, about how you take your samples. So if in this part of the world we band apply our fertilizers, what that means is that the vast majority of the soil isn't directly in contact with that fertilizer, and there's a very small zone that's much more concentrated. And over the growing season, the phosphorus leaches out and dissolves and is taken up by plants. And so if you were to flag where you put the phosphorus in the soil, you could go out and with a sophisticated tool like a shovel and a piece of plywood with some nails in it, you could go and collect that soil sample. You know where the phosphorus was initially, and you could look at what happened to it in the field. And you can, at that point, the area where the fertilizer ban was is going to be weight percent phosphorus. You can measure away from it until you hit the detection limits, and then perform speciation. And so you could see that down here on the bottom right, where we see that the quantity of phosphorus goes from a fairly high concentration right in the band to a much lower concentration as we get away from it. And the speciation is also changing as we move in distance. And so what's happening is that fertilizer, as it's applied, starts to dissolve some of the carbonates that are in the soil, reprecipitates out its calcium phosphates, and kind of there it sits. And then over time, that calcium phosphate is going to then be dissolved and taken up by plants. And so you can kind of look at what is the fate of this thing over the course of a growing season by taking those focused measurements of what the concentrate is like. So yeah, so you can either choose easy systems. That's probably the recommendation number one I would give you. And if you can't get away with that, then you can try to remove some of the uncertainties in your reference libraries. That can be relatively straightforward, where, if you wanted to look at adsorbed phosphorus in a particular part of the world, then you could look at what minerals are there that are likely to be the sites for phosphorus to adsorb, and make your reference materials out of those minerals. The other thing you could do is start to look at what are the things that what are the kind of solid phases that phosphorus might form at a particular place. And in this part of the world, I think it's kind of underappreciated the fact that we have a lot of calcium, and the soils have a lot of calcium carbonate, and calcium phosphates are important, but that there's at least as much magnesium in many of these soils as there is calcium. And so you can improve your reference libraries actually by looking at what happens when phosphate forms mineral phases, not just with calcium, but with calcium and magnesium. And so if you systematically vary the pH of the system, that ratio of calcium to magnesium, you can synthesize a large number of materials. You can look at their XANES spectra, and you can also kind of figure out whether those are more reasonable for your system that you're studying or not, compared to things that you can buy as a reference material from a mineral library. And when that's done, you actually could see dicalcium phosphate, that brucite, which is a fairly poorly crystalline material, actually has the ability to incorporate a large amount of magnesium in its system without its structure changing too much. And when that happens, it certainly becomes even less crystalline. You see here in the green region is kind of that intermediate phase. The X-ray diffraction suggests that there's a fair amount of amorphous material in here, but that whole system is dominated by exactly the same scattering peaks. So the green spectra here in the middle are the same phase as the orange spectra. They're just much less crystalline. And then when we get to very, very high magnesium concentrations, then what happens is that we make another phase, which is isostructural with brucite. It's just the magnesium HPO4. So we make a magnesium phosphate at high levels of magnesium. Over the majority of the mixed calcium magnesium regions, we make a brucite phase that has substitution of magnesium. And then at really high levels of calcium, then we'll make the pure phase. So this implies that maybe what we were using as our reference material had a lot more structure than it should have. And already we can do better in speciation if we're picking minerals that are more likely to form under the geological conditions that we're working at. The other thing that we learned out of this system is if you go to a higher pH and you look at apatite as your phase, that apatite either will make amorphous calcium phosphate, which still seems to have largely the apatite structure. And so the implication is that amorphous calcium phosphate is actually just nano apatite, just very, very small particulate size apatite. And that's a transient phase that turns into apatite fairly rapidly. And then it takes really low amounts of calcium in that system to make the magnesium analog, which is a weird mineral named bobrite. So these are both the same structure. It's just we're replacing calcium for magnesium. But at those margins, when it's a really crystalline calcium phosphate, the mixed solid phase just doesn't happen. You make a small amount of bobrite and a lot of apatite, or you make only apatite, or you make only bobrite. You don't really incorporate magnesium into that structure. It just doesn't fit. And so given that, we actually had an experimental system that we could investigate where we thought it was quite likely that these mixed calcium magnesium phases might form. And the reason that we thought that was because we were doing a field trial where we were injecting phosphate and magnesium and letting reactions take place in sort of little controlled microcosms that we placed into the groundwater. So the microcosms are in contact with soil that were from the contaminated sites. They are getting bathed in a magnesium phosphate sulfate solution and also hydrocarbons. And so they're undergoing normal bioremediation, and we can kind of look at what happens to the mineral phases of phosphorus. And when we did that, what we found was that if we manipulated the samples by getting rid of the carbonates before we started the experiment, then we would basically form magnesium phosphate phases. If we didn't, we formed a mixture of this magnesium substituted brucite as the dominant phase. So unless you really, really get all the calcium out of the system, or if you don't put any magnesium into the system, this is the mineral that forms out there. So in fact, I don't know that anybody has observed this outside of our lab group, but it seems like that magnesium-rich brucite, which it's sort of about a 1-to-1 or a 1-to-2 ratio, seems to be the dominant mineral phase that forms in these systems. And once this was part of a bioremediation project, and what we were able to learn was you could see that the degradation rates and the enzyme activities of degrader organisms had a reasonably good fit to the quantity of adsorbed phosphate that was in that system. So if you can push that geochemical condition to try to stimulate adsorption rather than making minerals, then you can stimulate bioremediation. So that also means then that the microbes that are growing in this system, they can directly access and grow and metabolize the phosphate that's adsorbed onto the mineral surfaces, which is also a new finding. Okay, so what we've gotten so far is you can pick a really easy system. You can make better references and then pick a system that you expect to find them. The third option is we can get fancy and try to cruise around in our sample with a microfocus beam and directly measure the individual components. So as the beam gets smaller and smaller, it's more likely that a hotspot is only going to have a few types of phosphate. And so if we can do speciation at those spots, then we can do a better job of understanding what our reference material or what our components should be. You can already see that we've gone from 2002, where we were studying sort of 3 to 5 weight percent phosphorus manures, to soils that were kind of reasonable concentrations, hundreds to maybe 1,000 milligrams per kilogram, to now we have a microprobe beamline that can work at this sort of phosphorus energies, which is a fairly recent advance in the spectroscopy. And so here, again, we had samples that they were soil samples that had initially had different forms of phosphate added to them. And then we could go back after 3 months, 6 months, or a year and look at how the speciation had changed. And so we're able to look at what the transformations and the fate of these different forms of phosphorus are over time. So, there was a biochar that was made from the bones of fish. There was a biochar that was made from the bones of cow. There's tripolyphosphate was added, or just orthophosphate like NPK fertilizer, or no phosphate was added at all. And you can see that, we still had, 1,000 milligrams per kilogram phosphate even in the control sample. And that's one deal with phosphate, is that, if it hasn't been taken up by plants and it's a situation where it hasn't leached, then it's going to be persistent. So your control or your blank may still be above detection limit. But you could see over time, this is 2,500 milligrams per kilogram down here for the polyphosphate, and much higher concentrations even for some of the biochar compounds. We can do linear combination fits. We can get a good fit and a good summary of what's going on in the system. But again, is it right? Is this really the unique solution? And so what happens if we actually cruise around in a microprobe and look at some of these samples? I'll point you to the fact that the vast majority of the phosphorus in this poly sample is adsorbed, and in the biochar sample, the vast majority is calcium phosphate. So then if we do mapping of the elemental distributions or of the samples, you could see, these hot spots of calcium. You could see areas where there's silica and iron, but the phosphorus is actually fairly evenly distributed through the whole sample. And that would be consistent if adsorption is the dominant mechanism. And so if you do a tricolor map or a false map and you overlay where the phosphorus is in green, where the iron is in red, and where the calcium is in blue, you can see that there's some little hot spots where there's calcium phosphate, but for the most part, the phosphorus is kind of distributed through the sample. And then if we go to these different spots and collect XANES spectra, you can certainly see in some of these spots, there's some structure that is consistent with calcium phosphate, and in other spots that it's basically adsorbed. So our speciation model and our X-ray microprobe measurements are fairly consistent. In contrast with that, if you have materials that aren't very soluble, then as you go through the samples, you see a very strong co-location with the calcium and the phosphorus. And in some cases, you could still, though, collect spectra of adsorbed species. But what I wanted to highlight here is that when your samples are very concentrated in a microprobe, then the level of distortion from self-absorption and the changes in the the spectra, the artifacts that you get can be really bad at very concentrated samples, be very large. And so, if you were taking a sample and trying to do linear combination fitting on it from the microprobe, then it's very unlikely that you're going to be able to do that, given how distorted that some of these sample spectra are, just because there's so much phosphorus in that one spot. So as a qualitative approach, we can see that there must be crystalline calcium phosphate phases here, but we're running into that same problem that we had initially, where it can be hard to distinguish which is which. And that's not due to a lack of structure. In this case, it's due to self-absorption. Yeah, the other thing about this is that, it takes a shift or 6 or 8 hours or so per sample to be able to do the mapping on a reasonable size sample, and also then to do microxanes on a few spots. One thing that you can do with this approach that's really powerful is you could collect xanes at different energies too, right? So we could do calcium xanes, or we could do sulfur xanes or iron xanes. But every time that you add another element that you're studying, then again, you have to build another reference library. And then again, you have to spend the time. So that is going to be the major limitation of this focused work. And then the other thing is that you still can't really conclusively discriminate between the organic phases, the adsorbed, and the really poorly crystalline phases. You can, however, get some insights into if you think it's adsorption, what the likely phase is. If you see that it's a co-location with iron, then it's probably sorbing on oxides. If you see that it's calcium in this part of the world, it's either a very, very poorly crystalline calcium phosphate, if it looks like adsorption, or it's calcium carbonate that the phosphate sorbed on. And so you can start to make some inferences and start to improve your linear combination fitting of your bulk spectra with this approach too, and feel a little more confident that you're using the right phases in your models. The other thing you could do, and this is something that we're working on currently, is you could just try a different wavelength, right? Like, so you could, instead of using the phosphorus K edge and doing 1S spectroscopy, you could do 2P spectroscopy at the phosphorus L edge. And so this is done at the VLS PGM beamline. And this isn't a new idea. There was a paper about 13 years ago now, 14 years ago, that looked at characterizing a lot of the reference materials that you could potentially find in a soil, and using the MCP detector, a multi-channel plate, so it's total fluorescence. So you're measuring the signal from everything that's in the sample. And that has two problems. It makes the background corrections and the matrix that's in the sample difficult to normalize. And if you can't normalize even your reference materials, then you're going to really have a hard time doing any kind of quantitative analysis moving forward. I didn't know this, but chemists have a turn of phrase where when we say something is challenging, we mean that you should not do it because it's not going to work. And so I'm going to say this is a challenging baseline removal. And so we started doing this because of the fact that the selectivity rules are different. The discrimination that you get between phases is different. And as a result of that, you're a lot more sensitive to differences in organic phases, in adsorbed versus poorly crystalline, and you get a lot better, in principle, a better ability to speciate things like the chain length of polyphosphates and what that organic phosphorus is coordinated to than you do in the K edge. And so that's why we wanted to do it. It has to be done under a vacuum, so you always are going to be worried about changing the material when you pull a vacuum on it. The detection limits are kind of in that 2 to 3% range for historically. And the bigger issue here is that the penetration depth for soft X-rays at 100 EV, 150 EV is very, very thin. And so you always have to be worried under those conditions about a few different things. And I'll talk about some of those in a minute. But yeah, so I mean, people have attempted to do this. And large reference libraries have been published. This one here is in the journal Synchrotron Radiation, I think. And a lot of the work was done at PGM here at the CLS. When you get to a soil sample, though, this is just a constructed mixture of quartz with phytic acid, that organic phosphate that you might find in chicken manure, for example, or in a variety of grains. You see that most of the signal that you're actually getting in the total fluorescence is not due to the phytic acid at all, not due to the phosphorus oxygen or phosphorus. It's not due to phosphorus that's in the sample. It's due to the matrix. And so this is real problematic because it means that if you change the mineral structure, how much clay you have, how much sand you have, if you change the texture of your soil, which every soil is its unique little thing, then your background is going to change. And you also can see here in this range where the L edge is that there are some structures that overlap. And this is just a silicon oxide. If we had an aluminum oxide, it's going to be offset in a different way. If we start to add organic matter into that system too, we're going to have some real challenges in how to deal with this. Nonetheless, there's a paper that's been written on basically their approach was that they systematically diluted all of their references to exactly the same concentration in the same background. And by doing that, then they found that you could do quantitative analysis on mixture samples and that they could get a reasonable fit of concentrated samples like biochars and to the reference compounds. We tried this a long time ago. I never published it because I wasn't totally happy with the results, but it's fairly similar to what they did. Our approach was a little different. We used a liquid nitrogen and a grinder to get our particle size of our materials down to like 5 nanometers. And then with those very small, very fine, very well mixed samples, we could just do dilutions. And so this is a soil that had no phosphorus in it or very low phosphorus, and then increasing amounts of the phosphate material. This is what the raw data looks like. And then we just we subtracted, we moved the intensity at the low end to be equal to the sample and then subtracted it out as a ratio. And when doing that, you get a reasonably good calibration curve. And when you normalize all these, you get a reasonably good alignment. And so this suggests at least that there's not any big saturation effects, that the concentration doesn't have a big effect other than the data quality on the phosphorus compound. And that's probably because it's such a low penetration depth. Now, as I mentioned, this paper here by Huttart, I'm probably butchering their name, they actually published some of this work and they talked about how they did it. So they also came up with kind of a systematic mathematical way to pull the background out. You could read their paper for more details. I would argue it's still not very satisfying, especially compared to just only putting in a silicon drift detector and looking at only the partial fluorescence. So what I mean by that is, instead of taking all of the fluorescent signal, if we have the ability to measure fluorescence as a function of energy, then of emission energy, then we can use only the phosphorus as our ROI. And then when you pull the phosphorus signal out, you don't have nearly the baseline issues and the normalization problems. Now you can just pull your data into Athena or Larch, whatever you're using, and just fit a background below the edge, fit a background past the edge, and get reasonably good normalization without any splines and without any complicated removals or mathematical stuff. These are just single scans of reference materials. And so then, you do get most of the, you are able to access some variability in the poorly crystalline versus the crystalline carbonate or calcium phosphate phases. You do see a lot of diversity in the spectra of of other inorganic minerals. And you get the ability to do quantitative analysis or linear combination fitting because your background removal is no longer arbitrary. So this is a fairly simple system. We just made a constructed mixture of hydroxyl apatite, aluminum phosphate, and phytic acid. And so those all have fairly easy to detect features in them. But what is interesting is I gave Athena that entire library that you saw me showing normalization with, and it picked out these three phases, which were what the sample was made of, as the best fit. Now the concentration are not right, and I think that has to do with the fact that the penetration depth is so thin that you tend to bias it towards certain size classes of the particles. You know, if your particles are big and the beam happens to hit a spot that has that big particle in it, then it's going to overrepresent it in the fit. But nonetheless, being able to say something about what kind of organic phosphorus is there, or conclusively say that is organic, it's not adsorbed, it's not bruchite, is a big improvement. And I do feel that you can definitely do that. So what we learned at the L-Edge over the last couple of years of messing with the silicon drift detector is that self-absorption isn't really a problem. Surface heterogeneity is a problem. Using partial fluorescence yield is definitely the way to move forward. It still isn't perfect. I'm not convinced. We just made some measurements over the weekend. I'm going to try to see if you can actually see the difference between, for example, a AMP versus phytic acid versus lecithin. You know, can you do that kind of speciation in a mixture system or not? I'm not convinced of that yet. We'll see what the results show. But one thing I think is really powerful is if you can conclusively see what kind of calcium phosphate is in the sample through either K-edge or L-edge, and you've got both sets of data, then you can constrain the other set, and it's going to make your fitting better. So if you already know a lot about the phases that you can see with K-edge before you come to the PGM and do L-edge measurements, then your confidence in the fitting of the organics and of the poorly crystalline stuff is going to be a lot higher because you've constrained the other stuff. So you know, I think these techniques are really going to be very complementary to one another. And so yeah, in conclusion or in closing, you know, soil chemistry has I know it sounds like dirt. Maybe people haven't thought a lot about it in the past, but a lot of the grand challenges that face humanity are soil science problems or challenges. So, you know, food security, how are we going to make better use of limited fertilizer resources, environmental sustainability and water quality, and also reclamation and rehabilitation of contaminated soils. These are all research problems that have big implications for the survival of people. And similarly, they're very well suited to synchrotron techniques. And particularly as the synchrotron continues to improve its detection limits, its ability to measure samples not super pure and in a super strong vacuum, its smaller spot sizes, and its higher sample throughputs. I mean, these are all the things that everybody at the beamline is always working on. The more that improves, the better it links the molecular scale research, like I've been talking about today, to field scale processes that we care about. So with that, I think I have hopefully 5 or 10 minutes. I should thank the University of Saskatchewan for paying my personal bills, for INSERC for providing students and research funding, and the CLS for providing photons. And if you have questions outside of this, if you don't feel comfortable asking me or you think about it later, feel free to either hit me on Twitter, and I don't have a blue check mark, but that's okay, or at my email address. And if you have questions, I'm happy to field.
All right, thank you so much, Derek. That was super interesting. So again, if anyone has questions, if you're shy, feel free to drop them into the chat and I'll read them out for you. Or if you want to ask them yourself, there is that little raise hand icon in Zoom. So just throw that on and I can call on you. Mirnaz.
Yeah, hi. Thank you for the presentation. So I just have one question. I quite understand the issue with measuring total fluorescence yield for phosphorus LH. I know that for like with partial fluorescence yield, we are able to tune the detector and we are able to see exactly like to which level, to which state the electron is getting excited. but I didn't understand what's the issue with the total fluorescence yield.
Yeah, that's a good question. The big issue is that maybe 90% of the signal that's hitting the total fluorescence yield detector is not phosphorus. And so it's providing a background, but that background changes from sample to sample. So if you have 5 times as much phosphorus in your sample, then as a result of that, you're going to excite phosphorus more strongly, which means that the penetration depth of the X-rays into your samples is less. And so that's going to cause the background emission of silicon and aluminum and everything else, because you're sitting on their X-AS basically, to go down. And so it's not a solvable problem anymore. It depends both on, it depends in a positive way on how much phosphorus there is in this sample, and also in a negative way. And so it's just, we played a lot of tricks over 10 years with a lot of data, trying to deconvolute it. And the best way is just to not measure it.
Okay. Thanks.
All right, anyone else? And again, feel free to drop them in chat if you're too shy. So while we're waiting for other questions, I don't know if I missed it, but I was wondering if you could comment on some of the difficulties you've had with sample prep and stuff like that for such a soft element.
Yeah, I mean, well, one of the issues is that every beamline is set up a little differently. And when we first started doing these measurements, I did some of these measurements in the late 90s at Brookhaven, and it was in a helium environment, and you could measure liquid samples by just making a little Mylar bag, and not super complicated. You know, you seal it off. It had like a little baby thing that looked like a hair curler, but you make your little bag and you put it in front of the beamline. So you can measure adsorption samples with water present. You can measure liquid samples. No problem. Now, like SXR&B, you can measure under helium, but it's almost always under vacuum. And so you always have to be worried about people want to run as many samples as possible without opening up the vacuum. So you worry about cross-contamination when you're making them. You worry about, especially with a soil sample, you pull a vacuum on it for a while, maybe some of those particles, maybe water was holding it together. And so you've changed the structure. And then they fall off on another sample, and then you've cross-contaminated. So contamination is a big issue. That so self-absorption means that if you have to dilute your samples down, it's no guarantee with a mortar and pestle that you are going to make your particles all the same size. And so that's a big problem. And it's it's more of a problem over at the really soft X-ray beamlines like PGM, where your path length is so small. So you don't have your volume is almost all the area that's the size of the beam. That's not even to discuss the fact that there's probably in a lot of these samples radiation damage going on as well. And that's a problem. And for higher energy experiments, you can deal with radiation damage by cooling them down. But even at liquid nitrogen temperatures, maybe even liquid helium temperatures, I think the cross-sectional area there on some of these things is just so big. And then the fact that if you freeze your sample, you're not getting X-rays into it anymore. You're just measuring ice. Right? And so those are some of the problems. The other problem you run into is if you're trying to describe the landscape of Saskatchewan, and you get 20 samples to run over the course of your PhD, and those 20 samples are going to probe like 0.5 millimeter by 0.5 millimeters, how can you say anything meaningful? That, well, I don't always have the answer to that problem either. I guess you have to be clever about how you design your experiments, right?
Yeah, I can definitely see how something like that would be an issue, especially over such a large surface area. All right, last chance for questions, either raising your hand in Zoom or dropping something in chat. We'll give everyone a moment. All right, if not, then thank you again, Derek. It was really interesting to hear about the phosphorus in soils.
Yep, thanks for that.
Yeah, thank you. And for everyone else, next session or seminars, sorry, I'm getting a little tongue-tied now. Next seminar session will be Wednesday, May 17th, 1:30 to 2:30. And we will be hosting Robert Green. So tune in back for that in 2 weeks. Thank you so much. See you in 2 weeks.
Bye.